it gives me great pleasure to introduce Professor Samuel Ramsey, all the way from the United States. He was actually here in person a few years ago and told us about his work on varroa mites. So tonight he's going to talk about another parasite, which unfortunately I fear will be in the news a lot in the next few years. So this is new stuff about Tropilaelaps. So Samuel.
Thank you very much for that introduction. I am very excited to have this opportunity and would like to thank the sponsor that made it possible for me to put this onto my schedule. I would like to speak to you about the research that I've been conducting on what I'm calling the pollinator pandemic. In the same way that we had a virus show up in one region of the world and within weeks it was everywhere, we are seeing a similar situation with this concerning set of parasites and pathogens and things that show up in one region of the world and spread to our pollinators in other areas. And it always involves Apis mellifera, because that's the only honeybee species that has a cosmopolitan distribution and the capacity to pretty much set up shop anywhere. As a result of that, viruses, bacteria, parasitic organisms, any of them that really want to expand their distribution in the world have to jump on to Apis mellifera. Now, why are there so many parasites to begin with? I think is a question that not enough people answer when they throw you into a presentation about these organisms. I think we really need to think through the fact that honeybees have achieved something absolutely remarkable. They are some of the most successful insects on this entire planet, and that is pretty incredible to see. They've moved all over the world, and they have bent us as human beings to their will to make sure that they can be moved to all of these locations. And so one of the things that has drawn us to them so much is that they are incredibly productive organisms. They are able to create this structuring where their colony can have between 20 and more than 60,000 individuals all working together towards a common goal that allows them to collect a ridiculous amount of resources and store a huge amount of food, but not just any old food, food that is remarkably nutritively dense. What these bees have created in their colony is a concentrated system of both nutrition and a great system of thermoregulation and regulation of the colony itself. So if you were a small enough creature, one of the best places possible for you to live in this crazy biological world is inside of a honeybee colony. You may not know it yet, but you want to live in a honeybee colony. Their nests are the perfect home. They are perfectly temperature regulated, humidity regulated. There's food all year round, which is something that not a lot of locations can boast. The colonies are also remarkable in that even if you're not interested in carbohydrates, which is the primary source of food available in honey, even if you're not interested in pollen that's produced by these plants as a protein source, you can get a great source of fat from the bees themselves. If fats and proteins are what you really need in abundance, well, then parasites have figured out that these bees are a great source of very nutritively dense options for their food. And in addition to that, if you can find a way to get inside of the colony and stay inside of the colony, be in some ways accepted as an element of the colony's ecosystem, well, you are protected for the rest of your life. Most creatures don't want to tango with bees. And as a result of that, organisms that get inside of these colonies are usually protected from their own natural enemies, to the extent that if they make their whole life cycle about the colony, they tend to lose any capacity to have natural enemies because the bees are protecting that colony and everything inside of it from everything outside of it. And so when people ask, why don't we just find what controls varroa or Tropilaelaps mites in the wild and just bring that, like use that as a way to manage these organisms? And what it's rough to actually have to say to people consistently is, varroa doesn't have a suite of natural enemies. We have yet to find one, a single natural enemy that consistently attacks varroa mites. Now, we found generalists, creatures like pseudoscorpions that'll eat anything that's small enough for them to attack. But What you have to consider in that context is the bees aren't used to these pseudoscorpions. They won't allow the pseudoscorpions to police the colony and walk around and grab things off of the brood. That's not how they do things. The bees tend to very quickly recognize threat around them and to build out this entire system where they will use lethal force, generating an entire security detail for that colony that protects even their parasites from the concerns that they would normally have to deal with under typical circumstances. So that makes a honeybee colony evolutionarily an incredible place to live. That's also why the bees themselves have so many different mechanisms that they have developed evolutionarily to thwart interlopers, to keep other creatures from getting into their colonies. Because once those creatures get in there, they can stay, their populations can grow to huge sizes, and they can become really problematic parasites. And we're seeing this now, and I'm going to jump into that today. It's no wonder that the mysterious mite that you see running around on this frame, it's no wonder that these creatures have become so successful in such a small amount of time. Tropi mites have been parasites of honeybees, we believe, for less than 100 years. That is an evolutionary blink of the eye. We think it's closer to somewhere between 50 and 70 years. And these creatures have learned to exploit their honeybee hosts to an extent that is deeply concerning because it has happened so rapidly. Normally, when an evolutionary relationship of this nature develops, it takes, at the very least, hundreds to thousands of years to develop into some sort of stable balance. And these creatures have not gone towards a stable balance. They've just jumped right in and started destroying colonies. Normally, you cannot do that. Evolutionarily, that is not a favored technique for getting what it is that you want. But the Tropilaelaps mites are able to do this because of us. We are the problem. I hope that it does not hurt you too much to hear that. Maybe some of you already anticipated that would come up at some point in this, but in the wild, the system functions in such a way where the bees, when they're determining a new location for their colony, they will actually intentionally make sure that their colonies are spaced between 3 and 5 kilometers apart. That's about the largest distance that bees will fly when they're drifting away from a dying colony. And as a result, that punishes any parasite that is virulent enough to kill a colony because it won't be able to find another, and it will have written its own obituary. And so the bees themselves have actually evolutionarily structured a system that works for them typically very well in the wild. We, as human beings, have established these apiaries where there is always another colony right next to them, such that when one colony collapses, another colony can get whatever killed it. And that rewards the parasite rather than driving those genes that were problematic away from fixation. So, some of you are probably already thinking, that's not something that we're going to be able to change anytime soon. Nobody wants to have an apiary with one colony in it and then drive 3 kilometers to another apiary with one colony in it and just space all of your 1,000 colonies out in 1,000 different locations. That doesn't work. So because we have generated an unnatural system, we have to also have our own input into that system to make sure that that system can continue to function. I hear people say, why don't we just leave the bees alone? There was a time where the bees were just left alone. They protected themselves. They were able to live their best lives without us. And now that we are constantly intervening in the system, isn't that bad for them? Well, we intervened in the system when we took them out of their natural setting and put them in boxes next to each other. And now we have to intervene to keep that system going until we can get rid of the parasitic and pathogenic contexts that are making this so difficult for them to continue on. So the Tropilaelaps mite that you just saw on your screen a moment ago, Tropilaelaps mercedesae, is an organism that I've been referring to as an emerging threat to Western apiculture, and it is a pretty substantial concern. These parasites are from a genus called Tropilaelaps. There are 4 species, and none of them are easy to pronounce. As a result of that, I've been calling them the Tropi mite because there is no there's currently no accepted common name for this species. The only name for it prior to this point was Tropilaelaps. So Tropilaelaps mercedesae, Tropilaelaps clarii, Tropilaelaps coenigerum, and Tropilaelaps tai are the 4 species. The one that you will hear me discussing most consistently is Tropilaelaps mercedesae. This is the parasitic mite that's been spreading all over the world. It was originally a parasite of the giant honeybee Apis dorsata, which can be found in a lot of places in Southeast and South Asia. And then there was a host shift. There was a host shift because Apis mellifera was originally a rather fortunate bee. It made a great decision. This is the bee that chose that it no longer wanted to live in constant competition with the rest of the bees in Southeast Asia. Southeast Asia is like the cradle of civilization for the bees. We used to think that they originally evolved in Africa and moved into Southeast Asia, but that doesn't seem to fit with the data. And most researchers now believe that honeybees themselves, or the original common ancestor of honeybees, evolved in Southeast Asia and diversified. And that's the reason why that is the only region of the world that contains every single honeybee species. It is the cradle of civilization for bees in that tiny little region of the world. The reason why you will now see that Apis mellifera is the one that is just collecting all the different parasites and pathogens is because it left the area of the world where all the other bees were present. And that makes sense, to leave all of the competition, to leave all of the sharing of parasites and pathogens. But then when they got to Europe, one great thing is that they didn't have competition. They could evolve into this bee that produces more honey than any of the other bees. They could evolve into this bee that has larger colonies than any other bee. Normally, that would be suppressed in a competitive context. So it was a boon for the bees and eventually a boon for us. And that's why we moved them all over the world. The unwise thing that we did was move them back into Southeast Asia. Because when they left that region of the world, you have to remember that evolutionarily, organisms lose genes that they are not using. And so creatures that used to have to deal with a bunch of parasites and no longer have to tend to lose their parasite suppression genes. Creatures that used to have to deal with a bunch of pathogens and no longer have to lose their pathogen suppression genes. And because they left that area where there was just a high load of these parasites, they seem to have lost the vast majority of their parasite suppression genes. As a result, us moving them back into Southeast Asia has created this context where every parasite that sees them, if it can get into their colony at any point, it will be able to be incredibly successful and cause a huge amount of damage. And so we have to be aware that this is not going to stop. This is not going to stop with Varroa destructor. This is likely not going to stop with Tropilaelaps. There will likely be other incursions of parasites into the Apis mellifera colonies because of how we have moved these organisms around the world. Now, consider there is a map of the distribution of the Tropilaelaps mite that is outdated and has needed some changes for a while now. At the time that it was published, unfortunately, it had some errors in it that have really upset and confused people. And so the Ramsey Research Foundation has been going around Southeast Asia, documenting the different regions where these mites are present, so that we can create a much more updated map than the one from, I believe, 2003. So this is the distribution currently, as best we understand it, of the Tropilaelaps mites. Now, we suspect that these mites are also in Russia, Kazakhstan, Uzbekistan, and potentially Turkmenistan and Mongolia, but I have not had the opportunity to confirm that independently. I've talked to a couple of researchers in some of these regions of the world who have told me that these mites are present there, but we actually need to verify this. So I need to get some samples, see some samples, and verify the identification. The reason why I'm not quick to jump at that is because this map that you see above is the one that was originally on the OIE website. This is the organization for animal health. And so it's the organization that people tend to go to understand where threats to different organisms are present. This one was the only one available for Tropilaelaps for a while, and it showed that Kenya and Tasmania already have Tropilaelaps mites. These were misidentifications likely of pollen mites, but they did drive a fair amount of concern in both of these regions and created this sort of false positive that established a boy who cried wolf sort of feeling about these organisms. And we don't want that. And so I've been working on being very circumspect about these declarations and making sure that we are doing the best possible work necessary to identify these organisms and be sure of those identifications. But what you can see is that these parasites have spread quite a bit from their original location in Southeast Asia. This was the only region of the world where these parasites were present. So Thailand, Burma, Laos, Cambodia, some regions of the Philippines, Malaysia, and Indonesia, this region of the world. And then they began to spread. So they've moved outside of Asia now that they are officially in Papua New Guinea, now that they are officially in Iran. They, of course, moved into South Asia and East Asia, the two regions of the world that they moved to first, and have become a huge problem in Korea. They are an issue in some areas of southern China, and definitely have created some problems in South Asia, India, Nepal, Bhutan. And then they've moved into the Middle East. And this is the part of things that really makes me nervous on your behalf and on our behalf here, because they're in primarily the eastern part of Iran, but it doesn't take long for these organisms to move into other regions. When they move all the way over to the western sections of Iran, when they are on the border with Turkey, then you guys, if you don't currently have an emergency response plan in place, this is the time to develop one, because Turkey is that location that's likely going to allow for more distribution into Europe and other regions of the world. It's kind of that bridge there. So the other thing I want you guys to make sure that you are noting here is the parallels with Varroa destructor and the likelihood that you will continue to see these parallels in the future. Now, I don't have a crystal ball. I don't have the capacity to actually say what the future will hold. And so there are some people who would say, why are you going through all of this work trying to understand this Tropilaelaps mite that's not in the Western world? It's still only in the Eastern world. Well, we can look at trends from the past and see what will likely be in the future. So if we look at the trends of the past, if we look at the fact that originally in 1904, when Varroa destructor was discovered, it was only present in this region of the world, Southeast Asia again. And then about 50 years after it was discovered, started spreading into East Asia and South Asia. And then after spreading into East Asia and South Asia, it then began to spread into the Middle East. And after spreading into the Middle East, moved into the Western world. It eventually moved into South America and North America as well. And after it spread, so after it reached the Middle East, there was only about 15 years before it was seen all the way over in the Americas. After it reached the Middle East, there were less than 10 years before it was seen in Europe. And so the Tropi mites have been in the Middle East, confirmed in the Middle East for more than 5 years now. And so it's important to recognize the ways that these organisms are capable of moving, and the fact that we have done a lot to maximize international travel. And as a result of that, these parasites are able to spread much more quickly. Australia was the region of the world that was able to keep these parasites out for the longest. The fact that they are isolated really allowed them to do that. And they just got these Varroa mites in their colonies in Australia. And I'll actually be heading to Australia in a couple of weeks to talk to them about what can be done at this point. And the thing that really allows such dramatic spread so quickly is because the mites have evolved the capacity to have a single founder establish an entire population. When a female Varroa mite emerges from outside of the cell capping, she is already pregnant, already fertilized, and that one mite can be moved anywhere in the world where bees are and establish a new population. And so, like in Reunion Island, a region that was free of Varroa mites for a long time, and around 2016, these parasites became established on the island. Well, it's possible that it was just a single mite, because we know the point of introduction, we know the area and the colonies, and it seems like, and it actually could be the case that just a single mite was able to accomplish this entire population explosion that you see here. So this pollinator pandemic, as I've described before, has a lot of similarity to things like COVID. And we have to recognize that if we had spent time studying COVID before it distributed itself around the world, we wouldn't have had to shut everything down, or at least definitely not for quite as long. And we would have been able to mitigate these issues much more effectively. But instead, we waited until the building was on fire in order to start writing down an emergency response plan. And we cannot do that again. We are in a similar situation with Varroa, where we definitely in the US waited to study this organism until it arrived on our shores, and that allowed it to become established and create a huge amount of problems before we were able to really figure out much of anything about it. Now the same thing is happening as the Tropilaelaps mites are moving. It's possible that they will never arrive. They may never arrive in Europe. They may never arrive in the US, but it would be foolish to consider that possibility and decide, well, since it's not here and it's possible it'll never get here, let's never develop a plan. We have to develop a plan. And I've been told on multiple occasions by more than one funding agency that there's a fairly small amount of money being divided amongst bee researchers. We have to prioritize certain things. We're going to prioritize the parasites that are actually here rather than parasites that aren't here yet. And it's the yet that's really fascinating, because a lot of people seem to be under the impression that it's only a matter of time, but we should wait until they arrive in order to study them. And I do not agree. So let me tell you a bit about what we know of these parasites. Tropilaelaps mites are smaller than Varroa mites, which makes them more difficult to see. They're about a third the width of Varroa. They create multiple feeding wounds when they are feeding on their host, and these feeding wounds are special. They're very different from Varroa's feeding wounds. When Varroa feeds on a host, it bites a hole in this region, and something about its salivary contents seems to keep this hole from closing. So it and all of the other mites in that particular brood cell feed through that single wound. And it seems to be an evolutionary mechanism that is developed to make sure that they don't cause so much substantial damage to the developing host that the host dies, because if the host dies, they die. They can't chew their own way out of the cell. Well, the Tropi mites have not had time to develop that sort of more balanced evolutionary relationship. And so instead, they run around and bite a new hole in the body of the host every time they feed. Bacteria are able to get into these wounds. We're not sure what exactly the mechanism is for it, but when they bite these holes into the host, and you can see we've stained the different wounds on the bee such that they're blue so that you can see them more easily. This is from a mite that was only present with the host for less than a day, and these are all the wounds that it's caused. Each one of those wounds has the capacity to develop something like scar tissue, such that that region of the bee's body, whatever it's destined to be, ends up being immobile and kind of bent in a particular way. And so you see this with the antenna of the bees. This kinked region that you see here is from an area where it was fed on, and every section after that is then immobile. So they can no longer flagellate their antenna the way that they would normally be able to when they're healthy. And you can imagine what kinds of issues that would cause if they feed on a region that's destined to become a wing. If they feed on a region that's destined to become a leg, you'll sometimes see bees walking around in colonies where regions of their legs are immobile, and this is very likely a result of the Tropi mites feeding itself, not necessarily viruses, because normally when we see deformed elements of our bees when Varroa are in the colonies, we attribute that to deformed wing virus and the result of viral infection inside of those bees. That doesn't mean that the Tropi mites don't have the capacity to also transmit viruses. They transmit multiple viruses: deformed wing virus, acute bee paralysis virus, and very likely black queen cell virus. That's only 3, and Varroa has at least 13 that it's capable of potentially vectoring to these bees. But the Tropi mites are just not as well studied as Varroa, and it's very possible that as we look at the other viruses that Varroa transmits, we'll find that the Tropi mites are capable of transmitting the same ones or newer ones. So we know that the populations for these mites grow very quickly, much more quickly than Varroa, at least twice as fast as Varroa, because of the fact that these parasites have a much shorter phase on the adult bee population. It seems to be a true phoretic phase rather than what Varroa has, which is a pseudo phoretic phase. The varroa mite pseudophoretic phase is considered as such because they do feed on the adult bees while they are on their bodies. And phoretic organisms are organisms that value their host so much as a mode of conveyance that they don't feed on them, they don't cause damage to them, and they don't use them for reproduction. They just use them to take them from one location to the next. Tropi mites spend very little time on the adult bee population, time as measured by hours rather than the close to 2 weeks that you'll see with varroa. And that makes sense for an organism that's feeding, spending a lot of time, versus an organism that's not feeding, spending very little time. We also know that Tropi mites don't need honeybees quite as much for movement. They're able to run very rapidly and run substantial distances that you don't see with varroa. So we know that their populations grow quickly. We know that the colonies collapse quickly as a result of these parasites. But at the end of the day, what I've detailed to you in the last 3 slides is pretty much the full complement of our knowledge about these organisms. There's not a lot of things that we have very clearly worked through in the lab and understand about these creatures. And that is a huge concern, because at this point, we know enough to know that we need to know more. We know enough to know that we don't know enough. So there are questions that I've posed about these organisms that I think desperately need to be answered. I'm not a territorial researcher. If you or anybody that you know wants to tackle any one of these questions, please jump right in. These are not questions I've locked down as what only the Ramsey lab is jumping into. But we do need to know what these parasites eat. When I first started working with them, I was told that they fed on the bee's blood. I was like, oh, great. I'm really glad to hear that you guys actually know that from experience, because we had this crazy situation with thinking that the varroa mites fed on the bee's blood. It turns out they didn't. And they're like, oh no, we believe that the Tropi mites feed on the bee's blood because the varroa mites feed on the bee's blood, and they're very similar. That was the reason why I started disseminating my work, making sure that it's more easy to disseminate in other countries as well. It's one of the good things about publishing open access too. And so they are aware now that we don't actually know what the Tropi mites feed on. Those assumptions were not helpful. So we need to know if they're feeding on the hemolymph of the bees. We need to know if they're actually feeding on the fat body like varroa. We need to know if it's something else entirely. And at this time, that information is just not something that's been determined. Do they feed on the adult bee population? We have assumed, because they spend so little time on them, they don't. But once again, that is an assumption, and I am tired of us assuming things in the world of bees and then basing entire systems off of these assumptions. That is a very, very bad idea, and something that we absolutely need to stop. What does it take to rear these parasites in the lab? A lot of the questions we want to answer about them are questions that would best be answered in a laboratory setting, but no one's ever reared these parasites in the lab, and it's going to take some work to figure out the ins and outs of it. It took some work to figure out the ins and outs of rearing varroa in the lab, and it's been almost 60 years since the first studies attempting to do so, and it's still very difficult to do. And then how do they spread? How do they get from not just one colony to the next, but one country to the next? If we can figure out and best understand their methods of spread, we might be able to mitigate it before we have to deal with them all around the world. And then finally, what methods of remediation actually work to control these organisms? So there are multiple ways that parasites of this nature can be remediated. We know already from dealing with brood parasites like varroa, where things like formic acid have been effective, amitraz, but what options will work against the Tropi mites? Will heat treatments work? Will brood breaks work? Should we try the formic acid and the amitraz and oxalic acid and all of these other options that have been typically made available for varroa? A lot of the work that needs to be done to understand the Tropilaelaps mite is observational work. What happens when these parasites are with their host is an observational study. We need to look at these things and understand exactly what's occurring and take really clear notes. And that's really difficult to do because, unlike varroa, the vast majority of their life cycle is under the cell capping. Varroa has about a 50-50 split of a bunch of time on the adult bees and a bunch of time in the cells, sometimes even more time on the adult bees than in the cells. The Tropi mites are almost always inside of the cells. That makes an entire section of their life inaccessible to us because it's in this opaque context. And the only thing that we can typically do under those circumstances is open the cell, look at what the mite was doing at the exact time that we opened the cell, but know that now that we have opened this cell, we can't actually know what would have happened after that, because we've changed the behavior of this organism by removing it from its normal context. What we would really need to be able to do is see these parasites inside of the capped brood cell, even though it's pitch black and very difficult to see. That would be the best context for better understanding them. So the Ramsey Research Foundation, partnering with National Geographic and now my university, the University of Colorado Boulder, have actually started multiple sets of studies now to better understand what exactly is happening with these parasites, multiple kinds of parasites. So this colony that you're seeing here is a colony of Apis cerana bees. Digging around in there, trying to get some samples, not just of the bees themselves, but also of hive debris, which can tell you a lot about what organisms are living in the colonies with the bees. And what we have consistently noticed as an element of all of this is that climate change has played a lot into how all of these things have been working. It's played a lot into human encroachment. As the climate changes and people have to move away from the coasts and more into forested regions, a lot of times they end up putting a lot of stress on the bee populations themselves. A lot of these bee populations don't want to be in consistent contact with humans. So originally we thought that problems like the spread of these parasites were going to be climate blind because they're being spread by the bees themselves and not so much through climate change directly. But as these bees become more stressed, the wild bees, the Apis dorsata, the Apis cerana, the bees that you see nesting in logs and on trees, when they become more stressed, parasite populations grow much more quickly in their colonies. And when they have more parasites, but are also interacting in areas where now we have brought Apis mellifera into it, there are more opportunities for host shifts. And that is the reason why it's become such an imperative for me to go to these regions of the world and actually collect every species of honeybee out there. So my goal was originally to just collect Apis dorsata and Apis mellifera and look at the Tropi mite populations. I've done that now. And now that Nat Geo is partnering with us, I have the capacity to do something a lot more ambitious. So I'm going to every region of Southeast Asia where there are honeybees, and that's pretty much every region of Southeast Asia, some regions of East Asia and South Asia as well. And I'm going to document every single species of honeybee, every single parasite and every single symbiont that these organisms have. This is me in one of the jungles in northern Thailand. But I've also on that same trip gone to Bangalore, India, and Nepal and Singapore. Next up are the Philippines and Nepal next month. We are heading to Korea and Japan next year, as well as Vietnam. And we still need to figure out a good collaborator for Cambodia, Sumatra, Borneo, Sulawesi, Mongolia, and Taiwan so that we can get samples from them. At the end of this project, I will have found all 9 species of honeybees, the 2 subspecies that we think may actually be separate species themselves as well, so 11 altogether. And we will have documented all of the symbionts that we can possibly find in their colonies, all of their pathogens, such that if there is some sort of outbreak of one of these organisms in the world that is attacking Apis mellifera honeybees, we will understand it a lot better. I've been calling it the melitophiles because a melitophile is an organism that loves to live in honeybee colonies. It's from the Greek word for honey and the Greek word for lover, melitophile. And so it's kind of a play on words because I like puns. In studying these organisms, it's actually yielded a lot of data specifically about the Tropilaelaps mites at this point. And I would like to tell you guys a fair bit about what we have found in that time. So I've already discussed that when stressed out populations of bees are moving around, they can distribute their parasites. So we know with migratory beekeeping, which is a much bigger thing in my section of the world, when we have a bunch of stressed out bees in Florida, and then we move all of those stressed out bees to California, we create this system where we're moving all kinds of pathogens and parasites to another region of our country, and it allows for a cycle of disease to move around. And so we try our best not to move the stressed bees. Well, in Southeast Asia, Apis dorsata and Apis cerana are more likely to begin migrating early or to move to multiple different locations when they are stressed out. And that allows them to just sprinkle parasites into the population, which is why you see me and my postdoc looking at comb from Apis cerana bees to better understand what parasites they have there and Apis dorsata. Now, what you're looking at, if that video is coming across, hopefully as clearly as I would like for it to, you're seeing the very first video ever collected of a Tropilaelaps mite inside of a sealed brood cell with its host. That brood cell is not open, but you are getting a chance to eavesdrop on what exactly is occurring there. The study that I conducted of trying to understand the Tropilaelaps mite was divided between two segments. We wanted to be able to see what the parasites were doing for observational context, and then we wanted to be able to actually kill the parasite. And so we had it separated into the cite the mite study and the fight the mite study. And I really appreciate just how much the beekeeping community rallied around this work and made sure that it could happen, because when it was originally considered a lot more risky and more of a risky bet to actually fund, beekeepers were the first people to fund this study. They raised nearly $100,000 to make sure that I could go to Thailand and study these parasites with a system that I literally developed in my friend's garage. We created this cite the mite, what we call Mite Insight system. And so the fight the mite and cite the mite sections of the study were actually really successful, which I'm really excited to say, because originally they were not. When I first went over to do this study at the end of 2019 and got everything set up, you might be able to guess what happened at the beginning of 2020 when the project was set up and ready to go. And I was about to get all of this done. And then COVID hit and we thought, oh, well, maybe we'll just be gone for a few weeks. But those weeks turned into months. Those months turned into more than a year. All of my colonies got destroyed by giant hornets and other issues. And I wasn't able to do that work. And I felt absolutely terrible because this was an investment of a lot of people. But then Project Apis M and the United States Department of Agriculture stepped in to provide the funding needed to get everything started a second time. And as a result of that, we were able to get some really incredible observational data about what these parasites are doing inside of these colonies. We figured out a lot about Varroa through inference and through trying to understand, like looking at them as we open these different cells and peer inside what's happening with these parasites. But there's still a lot that we would have been able to better understand if we could watch them doing their business inside of the colony. So look at all of this fat body tissue that you see just bunched up around the aorta of the bee. The mite's mouth parts are pressed right into that fat body tissue. And we know a bunch of the issues that this feeding causes for the bees, all the elements of disease that it causes, the transmission of viruses, early desiccation, higher winter mortality. And at the end of the day, bees that have been fed on by these parasites tend to die more quickly. But none of that had been studied with the Tropilaelaps mite. And so this video that you're seeing now was one that I was just so excited that we were able to capture in high definition and watch these parasites doing what they do inside of these cells. The equipment necessary for this study was actually very rudimentary in the beginning. So you're seeing the garage setup where I melted wax, made these little thrones where 4 of the hexagonal walls, the normal 6-walled cell, are available here. And then the other 2 walls are replaced by this high optical clarity glass that allows us to peer all the way through the cell and see exactly what's happening here. And so you can see this is the very first time I did this study in this very rudimentary setup. After also starting at the university, I've had some nice startup funds to be able to infuse into this system and create something that is actually a bit more savvy than the original. But this windowing effect allows us to see directly into the cells, and then they're put inside of a special incubator that my friend and I designed in his garage that allows us to see straight through. And we can watch all of these different behaviors and then document what the parasites are doing throughout the entire nearly 12-day period that they are inside of that cell with that host. That kind of information is really important because we're eventually going to be able to develop it into a chart that shows you exactly what these mites are doing, how they spend the vast majority of their time, and weak links in their life cycle that we can potentially tackle for better management of them in the future. And while we're speaking of management, let's talk about fighting the mite, because testing potential treatment options was something that I wanted to make sure was a part of this project. I didn't want it to just be the kind of research that provided intrigue about these organisms, but also the kind of research that provided us actionable things that we can do to make sure that we are managing their populations. Now, as I explained to you before, the population of mites on the adult bees is the period of time that is the shortest for these mites. And that's different from Varroa. So with Varroa, we have a bunch of chemical treatment options that target the population while it's on the adult bee, because that is the period of time where Varroa is the most vulnerable. When they're under a cell capping, that cell capping is hydrophobic. Most pesticides can't penetrate that cell capping and get to them. So we target the population on the adults. We cannot do that with the Tropi mites. They have outsmarted that system. We have adapted multiple pesticides from Varroa to Tropi mites, and they have not worked. And my hypothesis was that the reason for this is because the vast majority of these insecticides that we have adapted only target the population on the adult bees. If we are going to actually target the Tropi mites where they are, we have to target the period of time where they've invaded the cell and are reproducing under the cell capping. And the chemical that is best capable of penetrating the cell capping and attacking populations of brood parasites under it is formic acid. And so I took multiple colonies, 20 colonies per each location in Thailand. We set them up in these regions, which I thought was going to be a lot more difficult than it was going to be. I was like, guys, we got 60 colonies. We're supposed to move all of them in one day. How are we going to do this? And they just broke out with this counterbalance that they just built overnight. And I'm like, why is everybody so much more resourceful than me here? I thought I was a pretty clever dude, but I wouldn't have made this overnight. Love it. Love everything about it. This brought me great joy. Anyway, so we got these colonies established in different areas where they had a good source of forage and a good source of water. This one is at Rajamangala University. This is Professor Guy Tanawat, who was integral in this work being conducted as well. And then each colony and frame received a unique identifying label that was affixed to the frame so that we could track what the parasites were doing to the bees and how that was impacting their ability to collect food overall, their productivity, and the levels of disease developing in them. Every single one of those frames, we captured pictures of them each week so that we could then compile a system where we could track the development of the brood, the food, and everything inside of the colony during that time. And you can see that we actually had to modify these colonies because the front entrance to these colonies is really, really, really small. Hornets are such a huge issue in that area of the world that the architecture of the colonies themselves is adapted to Southeast Asia. And so the group that I'm working with, Tanawat and M and Off, they were absolutely integral to this work being conducted. Formic acid was only one thing that we tested here. I wanted to make sure that we had other options aside from just using one chemical. Oftentimes when parasites like this arrive, because we don't have a good emergency response plan, we panic, we grab one thing that works, and we only use that one thing until it eventually drives a level of resistance. I wanted to make sure that we had multiple options. And so they're actually cutting open the colony now so that we can slide these commercially available heating pads into the colony that will heat the colony up to 106 degrees for 160 minutes to actually kill populations of parasites in the colony that way. So formic was one option. And these are colonies in which we have treated them with formic, and the bees don't want to be anywhere near those pads, but they're still inside of the colony, just chilling. But then we wanted to try what some of the beekeepers in Southeast Asia have been doing. Formic acid is not sold as a bee treatment in their countries. It is not licensed for that kind of sale, but there is formic acid available in a very concentrated form, typically used in the rubber industry. They will soak pieces of wood and then push those into the colony. And so I wanted to see how this method stacked up because it would be a lot cheaper and potentially a means that beekeepers could use resourcefully to protect their colonies from these mites. You can see I've got a lot of personal protective equipment on in that video because that stuff is very concentrated. And the bees don't like it at all. Like the vast majority of the adult bee population just boils out of that colony and runs away immediately. We haven't seen it cause the bees to abscond as the volume of formic acid in the air goes down. Eventually the bees start to go back into the colony, but we're not so worried about the adult bees. It's the brood that we want to use that formic acid against. And the brood is not capable of leaving the colony, obviously. The heating pad that heats the colony to 41 degrees Celsius for 160 minutes. You can see here that fairly large proportion of bees will ball up on the front of the colony in ways that you typically see when it's very hot outside. But something that's really interesting is that the queen is typically at the middle of this ball of bees. I think that's her attempting to preserve her sperm viability from these really high temperatures that could potentially impact the sperm. Some of the bees do die. You can see some of them are stuck to this board, but I also want you to notice that all of those red speckles that you see there, those are varroa mites that have fallen out of cells and died on the heating pad itself. So this, we have noticed, has had a fair amount of efficacy against the varroa mites themselves. And then one more method that we were trying here that was actually really exciting to see was a solar-powered system that was able, through a heating coil woven into the wax itself, able to heat individual cells that the mites are inside of. And rather than using a large amount of energy, and rather than heating the entire colony and destabilizing some of the context that the bees normally generate within the colony, they just heat the cells themselves to attack the mites. We had to establish this solar panel, which is not within my wheelhouse typically, but M, who you just saw walking across the camera there, is an engineer, and so he helped us get the solar panels all set up for our different apiaries so that we could use these systems in the colonies. We then did a frame-by-frame inspection of all of the brood, all of the food, how many bees were in the colony every single week, the week before we treated, the week after, and then all the way out to 8 weeks to see what impact this had on the colonies. These inspections were pretty thorough and involved a bunch of people all taking notes on these different qualities all together. And then we would take one frame at random that had brood on it back to the lab and open 100 cells and inspect the contents of those cells inside of the colony. And that was our way of actually noting how many parasites are actually present inside of the colony, because while an alcohol wash or a sugar shake would work under normal circumstances for varroa, you're already aware now that that's not going to work for the Tropi mites because of how little time they spend on the adult bees. And so we actually had to very carefully remove the bees from these cells, these developing bees, and look at how many parasites are present in the cells. If the colony had been treated, look at the impact that may have had. Are the parasites alive or dead? Are they now reproductively inviable? Are they reproducing? And all of that was actually very helpful for getting a clear indication of what's happening in these colonies. And you can see me being very shaky there with my forceps because I'm holding a camera, a light, and forceps at the same time, trying to get that image for you. So these were just our methods for doing this work. But I want to make sure I save time for questions. So let me get to the actual data itself. What you're seeing here is a representation of the data. All of these white hexagons are cells in which there was just regular healthy pupa inside. And if you see it replaced with the silhouette of a mite, these are cells the mite was inside of. This is the week before this colony was actually treated. And then after it was heat treated, you can see that the mites inside of the cells were actually dead. Every single mite that we found inside the cell for this particular frame was dead. And this was through the heat treatment, which was really fascinating. We also noticed a very similar system. So that very first week, we would notice a pretty substantial drop off with heat. Heat is the red line that you're seeing here. But then the issue that we noticed was that there was a rebound the next week, which was really confusing. We weren't always sure why that was happening in the brood. And it's something we're still trying to better understand now. We only saw that rebound, only ever saw that rebound with the heat treatment. You'll notice this purple and green line. The purple one is the liquid formic, the really highly concentrated, more dangerous formic. And then the more standardized system of applying formic that we use typically in the West. Both of those, the population of mites goes all the way down to close to zero. They just really get rid of, and this is Varroa in the colonies, but also Tropilaelaps mites in the colonies, and then it remains low for the rest of the study, rather than the staggered system that you're seeing with the heat treatment. And you can see that things are fairly consistent with the blue line, which is the control. Now, the mites inside of the colony, that if you separate that data out into just the Tropi mites, you'll notice that the population of Tropi mites drops precipitously from the very first treatment of formic acid and liquid formic. We actually didn't have to treat again, just used it once, and for an entire 8 weeks, that population was at 0 or close to 0. The heat treatment system required multiple usages and made it a bit difficult, but something that you can really see in the dead counts that we have here is that on week 0, so the week before any of these were treated, there are almost no dead mites in the cells, just a very small number, if any at all. But then after treatment with Formic Pro and liquid formic, nearly every mite that we found was dead, almost without exception. And it's the capacity of these chemicals to actually penetrate the cell capping and kill the mites under the cell capping that makes them a really useful option for potentially treating with Tropi mites, because we know of some of the things that can go after Varroa. There's a fairly dynamic blend of chemical options that can go after Varroa, but what we saw with Formic was really quite remarkable in its ability to penetrate the capping. The difficulty that we also saw is that it killed a fair number of bees. Both the Formic Pro, the standardized method, and the more concentrated method killed a fair number of bees and queens in these colonies. We were not able to use it by label application, specifically because in Thailand, it's very infrequent that you'll have an entire week with days below 84 degrees and humidity anything under 70%. And we know that Formic has a pretty substantial impact on the population of bees themselves when humidity and temperature are high. We noticed that as time went on, Formic Pro and liquid formic were the only options that continued to kill mites. We continuously saw by week 2 and week 3 that there was still enough of a concentration of formic in the air without a second treatment that mites were still dying under the cell capping. We did not see that with the heat treatment system. You can see here that the mites that we continued to find after that through the heating system were still alive, which is exactly what you expect. There is no residual effect of heat. And so our preliminary observations in this system is that repeated heat treatment did show something that was interesting. It seemed like the mites were no longer reproducing under those cell cappings. We need a larger sample size. We weren't able to find enough mites for this to be statistically significant at this point. And so we're going to conduct this study again. Now that I am a professor, I actually have postdocs and graduate students who can help with all of this kind of work. And I have an excited group of students right now and postdocs who are ready to jump into this project next year. So we'll be conducting this work again, but that still shows something potentially promising about heat. If it has the capacity to disrupt reproduction, it could still be an effective means, even if it must be coupled with other treatments to reduce the size of the population. It could still potentially keep the population from growing. Applications of formic appear to have the most dramatic impact on mite mortality, but also the most dramatic impact on queen mortality as well. I would like to thank everybody who contributed to this study, as well as High Flow and Manuka Doctor that also provided a substantial influx of funds to get this work done right before the pandemic occurred, and then there were issues there. And Project Apis M and the United States Department of Agriculture for making sure that this work could continue even after the painful blow that the pandemic originally dealt to us. And if there are any questions, I would love to take the time to answer them as that time is available.
That's fine. Yeah, thank you very much.
Okay, thank you.
Just while we're finding the first question, Samuel, in terms of your sort of emergency response, I'd just like to say that Victoria Soroka, who's here, has been in contact with a Kolos member in Russia, and Kolos is now launching a Tropilaelaps task force.
Good.
Gathering people, and that will be officially launched at the Kolos meeting in October in Slovenia.
Wonderful.
So I think the bee science community has been very complacent over the last few years, and while nobody's been watching, this thing has been spreading. So before it finds its way to the United States or Europe, hopefully people will be a bit more prepared. Right, and we have a question. So the question is, the sort of assumption that we've always made is that it can only live on brood and can't feed on the adults. And as you say, that's open to question. But how long can it survive without access to brood?
Wonderful question. We originally became very complacent about these mites because we didn't think they would be able to impact anything outside of specifically Southeast Asia. We didn't think that they could survive even in China because much of China gets very cold. And because brood isn't reared year round, the population of mites would just die off even if they were able to move into the country. So a very important paper on the subject of Tropilaelaps, like the title of the paper is, the Tropilaelaps mite, not going to be a problem outside of tropical regions. And we were surprised when this parasite moved into the coldest reaches of South Korea and China. These parasites can't survive for more than 2 days away from the brood, which they are capable of feeding on. So even if you just sequester them with adult bees, usually a little bit after 48 hours, every single mite is just dead. They don't seem to be able to feed on the adult bee population. So we aren't entirely sure how they're moving into these regions and how they're surviving in areas where there is a substantial winter, but they are doing it, and we have to figure out the answer to that question.
Thank you. Our questioner is from Poland, and he was saying that Professor Wojka, who sadly died recently, but did do some work on Tropilaelaps and reckoned that a brood break would control it. What are your thoughts on that?
Professor Wojka was right. Brood breaks are the primary means of controlling this parasite without chemicals in Southeast Asia. And most beekeepers that I know in SEA currently can't afford many of the expensive chemicals that could potentially do something. And so they do constant brood breaks. The fact that these brood breaks have to happen so quickly to avoid having the population of mites go out of control is something that I think of as a bit concerning, because remember, Tropi mite populations grow 2 to 3 times faster on average than Varroa mite populations. And so while a Varroa mite population can double every 30 days, a Tropi mite population can double every 7 to 15 days. And that is terrifying. So what beekeepers typically do in that region of the world is you'll very infrequently find colonies stacked the way that we have them in the US, where we have these giant colonies of 60,000 bees, the way that you guys do, of course, in Europe as well. In Southeast Asia, it's one box. Usually that box is only about half full. And then as the bees fill it out with brood, when they fill it all the way to the edges of that box, the beekeepers take the brood out of that colony and allow the colony to start over again. And it's this system of consistently breaking the brood cycle that allows you to keep these bees in the presence of the mites without the mites taking over and killing them. That dramatically reduces the capacity of that colony to build up. It dramatically reduces the strength and productivity of that colony, but it will keep that colony alive.
And the second question was, how do we think the mite spreads? Does it spread in swarms or colony movements, or what do we think?
Also a great question. Once again, this is work that needs to be done for us to really know this. Multiple researchers have speculated on this, and one of the books that I was reading called Beekeeping in Asia references that the Tropi mites don't hold on very well to bees in flight. And so it's unlikely that their primary means of spread is through drift or through swarms. I haven't been able to independently verify that. And so I wouldn't want to repeat that as if it's definitely a fact. And the individual who said this didn't provide any sort of experiment or anything to show that. But it is possible that Tropi mites, because of their increased capacity for locomotion, may just be able to walk right out of the front of the colony, down the leg of the colony, and up the leg of another into the next colony, because they do use locomotion as their primary means of getting between Apis dorsata colonies. Apis dorsata will typically nest all together, sometimes 300 colonies on the same gigantic tree, and the mites will just run back and forth between the colonies from one to the next. And we think that they're doing similar things in apiaries with Apis mellifera.
Gosh, that's definitely something to take account of that we're not used to. Right, we have a question from Fanny Hajina. So the second question first. Fanny's saying in tests with formic acid and Varroa, it was found that the sort of control was uneven and didn't reach to the bottom of the comb. Did you find the same in your tests, or was the kill of the mites even across the comb?
So thank you for asking that, because that was something that I also wondered about. The two different methods of applying formic. One, the Formic Pro that we used, we put those pads on top of the frames. But for the usage of liquid formic, we slid them in and they were under the frames. And I wondered if that would have some sort of different impact where the ones under the frames would have more kill with the cells that were closest to it. That may be the case in more temperate areas or with lower concentrations of formic. We used the highest concentration of Formic Pro that we could use. We used both little pouches of Formic Pro and the highest concentration of liquid formic that we could use. And also the temperature was very high. And when the temperature is really high, this chemical becomes more bioactive and is able to disperse through the colony more dynamically. And so while that's something that we expect we may find in lower temperatures and lower dosages, it was not something that we noted at all in this study. And your second question, I think, was about drones, if I heard it properly. Well, Varroa is definitely more attracted to drones than Tropi mites are. We expected that we would see some level of preference for drone brood in the Tropi mites, and we saw none. There was no greater amount of preference for drones in Tropi mites in the 2 years of study that I've done on this thus far. To justify what I've heard other people say about these Tropi mites also preferring drone brood, it just doesn't seem to be the case that they're interested. And in some of the colonies where we have had both drone brood and worker brood, we have seen a greater preference for them to be in the worker brood than the drone brood. It wasn't a statistically significant amount, and so I can't make that as a statement that is going to hold true across the board, but it is statistically significant that they were not going after the drone more than the workers.
Oh, that's really interesting. Another question in the middle there. Do they enter queen cells or not at all?
Great question again. Queen cells seem to repel brood parasites consistently across the board. And a big reason for this is because evolutionarily, you don't want to kill the one reproductive member of the colony, which will cut short your capacity to utilize that colony very, very fast. And so the full set of what we call semiochemicals, the chemical smell of the developing queen, is distinctive enough that evolutionarily they have developed an aversion to it. So the Tropi mites don't like to go anywhere near queen cells. Varroa mites will not enter typically queen cells. A few very odd mites, especially if they've had their sensory systems damaged, will go inside, but it tends not to be something that they are after.
So a question at the back there. So the question was about the heat pad and that you stopped at 41 degrees. Was that because you thought above that would injure the brood?
So I really wanted the heating pads to go hotter than that, because we know that the bees can deal with a higher threshold of heat than the mites can. And that threshold, the gap between them is multiple degrees. It's not like a hornet and a honeybee where there's only a 2-degree difference or so. And so I wanted that to go higher, but a lot of the studies of thermoregulation in bees have been conducted on adult bees. The brood itself does seem to be more vulnerable to heat. And in addition to that, one of my concerns is what impact does it have on the longevity and potential other health effects of these worker bees while they're developing inside of these cells if they're exposed to high heat and they don't die. But then what exactly happens? So we have had a little bit of time to conduct a bit of biochemistry on these bees, and they are different from bees that develop and aren't exposed to a huge amount of heat for a few hours. You'll notice that there's a large amount of something called heat shock proteins that develop inside of their bodies. They're upregulated, and a huge volume of these proteins is produced, much larger than you would ever see in a normal bee. These heat shock proteins keep the bee's proteins from unfolding when it gets really hot. They kind of scaffold the protein and push it back together and keep it from unfolding, which keeps the bees alive. But we don't know the long-term impacts of having a really huge amount of heat shock proteins all floating around in your body at the same time. So for that reason, we intend to do this study again, crank up the heat a little more, because maybe if the heat is higher, it'll actually have a more consistent effect on killing the mites. And then we are going to track biochemically how the bees change as a result of exposure to that heat, and then how long they live and whether that has any impact on their ability to forage and live their lives as worker bees. We don't know the answer to that yet, but we are working on it in the future.
There's just one more. We'll make this
Okay.
the last and then let you go. Did you hear that? How does it cope with Apis dorsata that migrates over considerable distances?
A really, really, really interesting question that we still don't fully know the answer to. To tell you the truth, I think that it is a crying shame that we have conducted so little research on honeybees in the region of the world where they developed. If you truly want to understand honeybee health, honeybee behavior, honeybee populations, you've got to do that work in Southeast Asia. That is the region of the world where the bees are being bees the most. And so these kinds of questions that seem like totally fundamental questions, many of them haven't been answered. A lot of the countries in Southeast Asia are considered to be, and I'm not a big fan of this term, but developing nations. And oftentimes the kinds of research that we conduct on bees is fairly expensive. So it can be difficult to jump into these kinds of studies. With Apis dorsata, we do know that they migrate every year, sometimes a couple of times per year, and oftentimes they utilize the same sites every single year. That colony will come back to that same region, even hosting themselves on the exact same branch. Well, something that is really interesting, and I want you guys to consider this for a moment because it was a mind blower for me when I first read this. Whenever I conduct a study on an organism that I've never worked with before, I always go back to the very first paper ever written about it and then trace my way forward so I can get an understanding of how we got to where we are. That's how I discovered that varroa mites don't feed on the bee's blood, but are actually feeding on the fat body. And that's also how I noticed the very first paper about Tropi mites, which no one cites anymore, actually cited these organisms as parasites of rodents. They were considered rat parasites for the first 9 years that we knew of them in science. And why was that? Well, because we were originally finding them on substantial numbers of rats in the Philippines and in China. And it seemed like that was their original host. And then we recognized that those rats were just very closely associated with bee colonies in those areas. Those rats were stealing honey and sometimes even clustering in or under those colonies. And I think it's actually possible that the Tropi mites may be using an alternative host. If they're not migrating with the bees because they can't hold on very well to the population of bees, they may actually be going into rodent burrows and living with rats. They may be living with squirrels or some other rodents that are very close in association with the colonies, that live nearby the colonies and even sometimes feed on the resources of the colonies. That has not been tested yet. That is still just a hunch, but I think there might be something to it, and I'm looking forward to verifying it. Not looking forward to being the guy who digs up a bunch of rodent burrows looking for these mites, though.
That's a fascinating possibility. But I entirely agree about the fact that, you know, Apis mellifera is just one of many species of Apis, and we know virtually nothing about all the other species. And when we had the Colos meeting in Thailand in 2020 that you came to and Vicky and I were at, you know, we were rather shocked by the lack of knowledge and the lack of concern about the other species of Apis, and that there were many beekeepers of Apis cerana who came to the second day of the meeting, but they seemed to be all of the view that Apis mellifera was much better, you know, really didn't know much about the populations of cerana. And we were taken to this Buddhist temple where there were supposed to be Apis dorsata nests under the edge, and you could see the cones there. And we were told, oh yes, there ought to be colonies there at this time of the year, but there aren't. And there seemed to be curiosity about why they were not occupied, you know, what's happening to the population. No knowledge at all, which is, you know, really serious. These things could be threatened, and we don't know.
Exactly. And that was something that was really concerning to me. There's a region of central Thailand. The city there is called the Bee Tree Town. And I love that. And right in the middle of the town, there's this giant tree where over 300 Apis dorsata colonies every year would come and land on this tree, build out a huge section of comb, produce a huge amount of honey. It supported the economy of this area. They sell Apis dorsata colony everywhere nearby. And for the last 4 or 5 years, fewer and fewer colonies have been showing up every single year, to the point that when I went last year, there were fewer than 90 colonies on that tree, a reduction that's pretty substantial and really concerning. and nobody knew what was going on, and no one is studying it. And it's a consistent theme that aside from Apis mellifera, honeybees just don't receive a substantial amount of study. So I'm really excited that National Geographic is partnering in this, and that I'm going to get to see every single species of bee before this project is over, because this is going to be a huge influx into our knowledge of honeybee species.
Right, well, thank you very much. We'd better let you go. So good luck with your ongoing research, and we hope we will be able to have you back here in a few years' time to give us an update. So
Looking forward to it. All right, see you, everyone.